Welcome to Layla's Science Hub. Today we're diving into one of the most important topics in biology, proteins. We'll look at how amino acids are structured, how they join, and how the different levels of protein structure directly relate to their functions. This is the basic structure of an amino acid, with an amine group, a carboxyl group, and an R group that changes for each amino acid. The simplest amino acid is glycine, with an R group that's just a single hydrogen atom. An amino acid is made up of carbon, hydrogen, oxygen, and nitrogen, and some also contain sulfur. Amino acids are amphoteric, meaning they can act as both acids and bases, so they can also act as buffers, helping to resist pH changes. When amino acids join together, they form long chains called polypeptides, and these fold into proteins. Proteins are huge polymers with diverse functions. They can act as structural components like muscle, adopt specific shapes to function as enzymes, or form channels and carriers in membranes for active transport and facilitated diffusion. The peptide bond is formed when two amino acids are joined together. This is called a condensation reaction, and water is another product as well as the peptide. It is called hydrolysis when this is broken. Protein structure is studied in 4 levels. Primary structure, the sequence of amino acids in the chain. Secondary structure, the folding or coiling due to hydrogen bonds between NH and CO groups. This creates alpha helices and beta-pleated sheets. Many hydrogen bonds together make these structures stable. Tertiary structure, the precise 3D shape, formed as helices and sheets fold further. This is held together by interactions like hydrophobic and hydrophilic interactions, disulfide bridges, ionic bonds, and hydrogen bonds. Quaternary structure, when more than one polypeptide chain comes together, like in hemoglobin. Let's zoom in on the bonds in proteins. Hydrophobic interactions push nonpolar R groups into the protein's center, while hydrophilic ones stay on the outside, especially in watery environments. Disulfide bridges form strong covalent links between two cysteine R groups. Ionic bonds occur between charged R groups. And hydrogen bonds keep tertiary and quaternary structures in the correct shape. Proteins can be fibrous or globular. Fibrous proteins are long, strong, and insoluble, making them ideal for structural roles. You need to know about these 3 examples. Collagen. It has a triple helix structure with many hydrogen bonds. It provides mechanical strength, found in arteries to prevent bursting, tendons, cartilage, connective tissue, and bones. Keratin contains many disulfide and hydrogen bonds, giving it great strength. Found in hair, nails, and fur, it provides mechanical protection and forms an impermeable, waterproof barrier to block entry of waterborne pathogens and pollutants. Elastin has extensive cross-linking and coiling, making it both strong and extensible. Found in skin for stretching, lungs for inflation and deflation, and blood vessels for stretch and recoil. Globular proteins are compact, soluble, and metabolically active with highly specific functions. You need to know about these 3 examples. Hemoglobin, composed of 4 polypeptide chains, and a heme prosthetic group containing an iron ion. It is a conjugated protein, meaning it has a prosthetic group, and its quaternary structure allows it to bind and transport oxygen. Insulin, made of 2 polypeptide chains held by disulfide bridges. It binds to glycoprotein receptors on cell membranes, stimulating cells to increase glucose uptake. Pepsin, a single polypeptide chain with stabilizing hydrogen and disulfide bonds, functions as an enzyme in the stomach, breaking down proteins into peptides. Exam tip: always link protein structure to its function. For example, hemoglobin's quaternary structure lets it bind oxygen, and collagen's triple helix gives it strength.